Okay, hello to all of you. Let's discuss chapter number 14. And this chapter number 14 is on genetics. So let's go into slideshow mode. So basically, chapter number 14 is about genetics. And in this chapter 14, we are going to talk about Mendelian genetics and the non-Mendelian genetics. And I have posted some videos about the Mendelian genetics and the non-Mendelian genetics. So Mendelian genetics is that genetics. Those are those laws of genetics which are given by by Mendel. I hope you must have studied in at your school time who Mendel was. So Mendel is considered a father of genetics, but Mendel was not a scientist. He was basically a monk, and he was doing some very simple experiments in his garden with using a very simple plant, peas. So, so basically, Mendel did not know anything about molecular biology or cell biology or anything about the cell division. So all the experiments which were done by him, they were done very simple cell pollinations and cross pollinations in his garden. So what remarkable about Mendel was that his insights were great. So he was just, growing some plants, crossing them, and he was his observations were great. So the most important thing is his observations were great, and his insights were great, and on the basis of those great insights he gave some laws. And at that time people did not agreed with him, and then later when people when the scientists they they found how the cell division works, what the cell division meiosis is, and what happens in meiosis, and when molecular biology developed and many advanced techniques were done in the labs, the science became so advanced, then people came to know that what Mendel was actually saying back in the past, he was correct. He was correct. He said everything correctly. And that's why he's called father of genetics. And he gave three main laws of genetics: law of dominance, law of segregation, and law of independent assortment. So in this chapter, first of all, we are going to talk about Mendel's monohybrid and dihybrid cross, and through those crosses we are going to understand the laws given by him. What is the law of segregation, what is the law of dominance, and what is the law of independent assortment. And then we are going to talk about non-Mendelian genetics. So non-Mendelian genetics are those genetic inheritance patterns which do not follow the laws of Mendel. So as we will proceed through the chapter, you will understand more. So at this time, so in the past, before the Mendel, there was a hypothesis which was very prevalent, and that was called the blending hypothesis. So according to this hypothesis, the genetic material from two parents blends together. So people or the scientists at that time used to think that when the offspring is born, the genetic information that offspring has, it is all because of the blending of the genes between the two parents. So that was the blending hypothesis. And analogy for the blending hypothesis is like if you have a blue and the yellow color, and if you mix that, you get a green color. So basically people used to think that the genes from two parents, they get mixed up. So the genes from two parents, they get mixed up, and then that mixture of the genes, that mixture of the genes is there in the offsprings. So this was the blending hypothesis at that time. But Mendel came up with a particulate hypothesis, and on the basis of this, parents pass discrete heritable units. So at that time, now we know what genes are, right? At that time, the word gene was not there, and the exact word which was used by Mendel was heritable factors. The word used by Mendel was heritable factors. So this is the hypothesis. Basically, came with Mendel's studies, and Mendel documented that a particulate mechanism through his experiments with garden. So he proved all these things through his experiments, which were done with a simple plant pea in his garden. So this is the plant, pea plant used by Mendel. So let me show you first. I will come back here. Let me show you the system of Mendel. So this is such a simple system of Mendel. So basically, he used scissors. He used the brush. Okay, he used the scissor. He used the brush. So with all these things, he was doing various cross pollinations and self pollinations, and um, he was working with like these are the pea pods. So these are the progeny. These are like seeds, and then or he was working with this is the pea, like the peas in the pea pods, and these are the plant of the pea. So basically this was a simple system he was using. Now um, he was using these peas, and there were many advantages, and like there are some characteristics of the peas which were like really were very useful to Mendel, and those advantages of peas are they have a short generation time, they have large number of offsprings, and also with the pea plants the mating can be controlled. You can do self pollinations or the cross pollination. So these are some of the advantages of using pea plants. Okay, so before I go and talk about the crosses of Mendel, so I'm going to in the first video for chapter number 14, I'm going to discuss the monohybrid and the dihybrid cross of Mendel. So basically, let's talk about these terms. So we need to know what a character is and what a trait is. So what is a character? It's a heritable feature. Character is a heritable feature that varies among individuals, such as a flower color. So it's a heritable feature that varies among individuals, like flower color, or it can be the color of the seeds or the texture of the seeds. So these are all features which differ among the individuals, and they are called characters. Each variant for a character, okay, each variant for a character is called a trait. Like if you talk about flower color, which is a character. Now the flower color can be purple or it can be white, and that is the trait. Okay, so flower color is a character. Whether the color is purple or white, that is the trait. Okay, another important thing to know about Mendel's pattern of inheritance is that he chose all the characters. Okay, he chose all the characters which come into two distinct alternative forms. Like the flower color is either purple or it is white. Okay, the seed color, either it is yellow or it is green. Seed texture, pea texture, either it is smooth or it is wrinkled. So he chose these kind of characters. So all characters are not like that. All characters are not like that. So this was another very important remarkable thing about Mendel's crosses is that he chose those characters which are there in the form of two distinctive alternative forms. And also he started with the varieties. Okay, whenever so in all his crosses you will see that he always started with the plants which are true breeding, or we can say they are pure. Now let me explain you what true breeding or pure means. So for that, let me I have to use a whiteboard for this. Okay, so now first of all let's talk about the chromosomes, the genes, the alleles, and then we can talk about what is pure breeding plants, or also the term homozygous and heterozygous. Okay, so as you know that in this chapter the knowledge of meiosis is very important. Okay, you really need to understand in this chapter the cell division meiosis, how gametes are formed. Okay, because in all these crosses and on your exam you'll get a lot of genetic questions. If you do not understand how gametes are formed, then it's very difficult for you to understand the whole process. Okay, so suppose this is your maternal chromosome. I'm making with red color. Now let me make the paternal chromosome with blue color. Okay, suppose here is the gene. Suppose here is the gene for the flower color. and at the same place is the gene on the paternal chromosome for the flower color. Okay, suppose this gene is P. Okay, in Mendel's crosses you will see that one of the allele, so the two variants of the gene on two different chromosomes, they are called the alleles. Okay, now let me tell you something about Mendel's crosses, like the monohybrid cross. So what's a monohybrid cross or what's a dihybrid cross? Monohybrid cross is that cross in which only one character is studied. Dihybrid cross is that cross in which two characters are studied. In the monohybrid cross, when the one character is studied, Mendel basically, I will show you a table in which there are different characters he has studied in different crosses, but we will talk about the flower color. So in the Mendel's monohybrid cross, capital P allele, so what is allele? These are the two variants of the gene. So the two variants of the gene on two different chromosomes, they are called the alleles. So the purple color is the dominant color. Okay, so purple color is coded by capital P. Okay, and the white color is a recessive character and it is coded by small p. Okay, now as you know that the chromosomes, they are in pair, right? There are two alleles for every character. Now this is the maternal chromosome, this is a paternal chromosome. If the allele on maternal chromosome is capital P, it means it is coding for which color? Purple color. And if this allele on this maternal chromosome is also capital P, then both the alleles on the two chromosomes, maternal and the paternal chromosome, are capital P and capital P. So the color of this flower will be purple because both the alleles are for the dominant character. So if both the alleles are capital P, just like here, this kind of a plant is called pure breeding plant. Okay, now there is a possibility, if this allele is capital P and this other allele is, suppose, let me make with different color, suppose this allele is not capital P but it is small p. If it's small p, then the two alleles are capital P and small p, right? But because purple color is dominant, capital P is a dominant allele. Okay, capital P, so the dominant alleles are written with capital letters. So if one allele is capital P, another allele is small p, then what is going to happen? Then this dominant allele is going to show its effect. It is going to express. So in this case also, this flower is purple. But there is a difference. The difference is, so this and this, they are both purple. This is, both the alleles are capital P. Here one allele is capital P, another allele is small p. Okay, both are purple, but there's a difference that this is a pure breeding plant, but this is not. This is also called homozygous. When both the alleles are same, that plant is, then it is homozygous situation. If one allele is capital P, another allele is small p, then this is a heterozygous situation. Now this, when both alleles are capital P, the color is purple. When one allele is capital P, another allele is small p, the color of the flower is again purple. So in both these cases, the phenotype, the external character is same, and that is purple. But their genotype is different. And what is genotype? Phenotype is the external appearance. Genotype is the genetic makeup. So in this case, the phenotype is same, both are purple, but the genotype is different. So in the genotype, both the alleles are capital P, but in this case, so this is pure or homozygous, but in this case, one allele is capital P, another allele is small p, so this is heterozygous. Okay. So I have explained you few terms here. Number one, what is the dominant allele, what is the recessive allele, what is genotype, what is phenotype, what is homozygous, what is heterozygous. Okay. So let's go back to the PowerPoint. And okay. So true breeding or pure breeding means the same thing. So basically, if you will see that in all Mendel's crosses, he started with true breeding or pure breeding plants, which means they were homozygous for the characters. Okay, so so he started with true breeding parents, and that generation is called P generation, which means parental generation, and the next generation is called F1 generation, and after the offsprings of F1 generation, they fertilize, they form the next generation, which is called the F2 generation. So I'm going to explain here. So this is basically Mendel's monohybrid cross. So what he did, he took purple flowers and white flowers and he crossed them. So purple flowers and white flowers, they are pure breeding here. So basically, which means that this is capital P, these are small p and small p. And then he fertilized them. And the next generation, which was formed, the F1 generation, all plants were purple in color. Okay, that's what is observed. And we are going to talk about how exactly is happening inside everything. So he took two pure breeding plants, one purple, one white. He crossed them, and in the F1 generation, he got all plants purple. Then he took the plants from F1 generation and crossed them, and in the F2 generation he got 3 purple plants and 1 white. So now let's see what is actually going on here. So he took purple plants with purple flower. The genotype is capital P, because they are pure breeding. And he took plants with white flowers. The genotype is small p and small p. Okay. Now, something very important to know here is he took 2 plants, and when he crossed them, so whenever there is a crossing, the sperm from one plant is going to fertilize the egg of the other plant. So sperms and eggs, they are gametes. So here comes the role of meiosis. So it's very important to understand, if you want to understand crosses, that how gametes are formed. Okay, I think I need to go back to the whiteboard and explain you. So let's see. Okay, so what Mendel has done, he has taken 2 plants, right? So one is capital P, another is small p and small p. So if you look on the chromosome, so this is one plant, okay, one chromosome is maternal, another is paternal. I'm just making with blue color, I'm not using 2 pens now. So these are the chromosomes, right? So here the gene for the flower color is capital P. On the other chromosome is also capital P. On this chromosome pair, in the another parent, this is small p, this is also small p. Okay. Now remember, because now what we are going to do, we are going to see how gametes are formed, because it's a gametes which fuse with each other, right? Sperm is going to fertilize the egg. So go back and see what happens in meiosis. Meiosis, in chapter number 13, we discussed, it has 2 phases, meiosis 1 and meiosis 2. So what happens in meiosis 1? It is the separation of the chromosomes. What happens in meiosis 2? It's the separation of the sister chromatids. So basically, sister chromatids are basically, they are the copies of each other. So basically what is happening in meiosis is the chromosomes are separating, right? So these chromosomes will separate and these chromosomes will separate. So from this parent, the gametes which will be formed, they will be capital P, this is capital P, okay. From this parent, the gametes will be formed will be small p and small p. So it's very important to understand that in meiosis 1, it is the separation of the chromosomes. And you need to understand how the alleles are there on two chromosomes and how the separation of these chromosomes takes place. And you can see here that this gamete will be formed. It will be having this chromosome with allele capital P, and this another gamete will be having the chromosome. This gamete will be also having the chromosome with capital P, and this gamete will be having the chromosome with small p, and this gamete will be also having the chromosome with small p. Okay, so this parent will form gametes which are capital P, and this parent will form the gametes which are small p. And these gametes, they fuse with each other, and next generation, which is going to be F1 generation, it's going to form offsprings which will be capital P and small p. So the genotype is capital P and small p, the phenotype will be purple. So basically in the parent, so I hope you know what the Punnett scale is. Let's make the Punnett scale for, so Punnett scale is like this. So one parent form gametes which are capital P, another parent form gametes small p and small p. Okay, when this gamete fuses with this, the offspring which is going to be formed will be capital P, small p. When this fuses with this, it's going to form capital P and small p. When this fuses with this, capital P and small p, capital P and small p. Okay, so all of them have same genotype. They are heterozygous and the phenotype is purple. This is F1 generation. Okay, we are talking about Mendel's monohybrid cross. Monohybrid crosses are those crosses in which only one character is studied. Now what he did, he took these plants, okay, so he got all these purple flowers. He took these and he made them cross again. So all the offsprings in the F1 generation are capital P, small p. So the gametes they're going to form will be capital P and small p. So how? Because on their chromosomes, so all of them have chromosome with capital P and small p. And these chromosomes are going to separate and they are going to result in the formation of these kinds of gametes. Now, if we do in the Punnett scale, so the gametes from both the male and the female parent are going to be capital P and small p. This is going to be capital P and this is going to be small p. So this goes with this and it forms capital P, capital P. Now we are talking about F2 generation. This is capital P and small p, capital P, small p. Now this is small p, okay? This is capital P. This is going to be capital P and small p. These are both small p's. Okay. This is Punnett scale for F2 generation. Now this plant is having both the alleles as capital P. So the color is going to be purple. What about this? One is capital P, another is small p. The color is going to be purple. Capital P, small p. Color is going to be purple. For this, both are small p's. The color is going to be white. So this plant is going to be white. This is going to be purple, purple and purple. So in the F2 generation, the phenotypic ratio is going to be 3:1. 3 purple plants with purple flowers, and 1 is going to be the plant with white flowers. This is the phenotypic ratio. Now if we look at the genotype, genotype is different, right? So basically this is purple, this is white, these are also purple. If you look at the genotype of this purple and this purple, this is homozygous purple, this is heterozygous purple, right? So if we look at the genotypic ratio, Mendel got one plant pure purple, two plants heterozygous purple, and one pure white. So the genotypic ratio is 1 is to 2 is to 1. So 1, this is purple, pure purple, 2 heterozygous purple, 1 pure white. So this is basically Mendel's monohybrid cross. Now let's go back to the PowerPoint. So this is what I just explained you. So in all this, you need to first of all understand the crosses will not be difficult for you if you understand how gametes are formed. And for understanding how gametes are formed, you need to have the knowledge of meiosis. You need to know in meiosis 1, it is the separation of chromosomes. In the meiosis 2, it is the separation of the chromatids. And chromatids are same, right? So basically, when you are making the gametes, make the chromosomes and write down the alleles on them and then separate them. And then you will be able to understand. So if this is a plant with purple flowers, which is a pure plant, this is the plant with white flowers, the pure white plant. So this is the genotype, capital P, small p. So these are the alleles on the chromosomes. During the meiosis, it will be the separation of these alleles, and that results in the formation of gametes. So for this plant is going to make all gametes will be having the allele capital P. In this plant, all the gametes will be having the allele small p. And when these gametes, when one of the gamete fertilizes the other gamete, in the next generation, which is the F1 generation, the plants which will be formed will be all will be purple because the genotype is going to be capital P and small p. And this is heterozygous purple. And when Mendel took these plants and he crossed them, so from this plant, the gametes will be, half gametes will be with capital P, half will be with small p. And when he crossed them, you can see this is what is formed. So this is Mendel's monohybrid cross. So this is one character he studied, okay? But in Mendel has actually studied many characters, okay? So these are all different characters studied by Mendel in his monohybrid crosses. So this was one monohybrid cross explained using flower color, purple and white. Then he also used in a monohybrid cross seed color, which is yellow and green, seed shape round and wrinkled, pod color green and yellow, pod shape, flower position and stem length. So basically he chose those characters which actually existed in two forms. Basically, if he chose the flower color, it was either purple or white. Seed color, it was either yellow or green. Seed shape, it was either round or wrinkled. So let's go back and see if we have explained everything. Yeah, so this is Mendel's monohybrid cross, and I have already covered what these terms means. What is P generation is the parental generation, F1 generation means the first filial generation, F2 is second filial generation. Okay, I will talk about law of segregation soon. So this we talked about. And at Mendel, when Mendel was doing all these characters, the gene word was not there at that time. So the word he used was heritable factor. So he said that the parents pass a heritable factor to their offsprings. We have already discussed what alleles are. Okay, we talked about what a dominant allele is, what is a recessive allele. We talked about what is a Punnett scale. We talked about all these things. We talked about homozygous, heterozygous, phenotype, genotype. So we talked about all these things. Now let me explain you one more thing here. Here. So basically, if you look here, so what does basically allele mean? So allele is a, so for every gene there are two variants and they're called alleles. So what is a gene? Gene is basically made of DNA. So you can see here, this is a paternal chromosome, this is a maternal chromosome, and this is the sequence of DNA on the allele which is for the purple flower, purple color, and this is the allele which is for the white. So you can see here, if you go back and see exactly what is there in the allele, because allele is a form of a gene. And what is a gene? Gene is made of DNA, and there is a sequence of the DNA, and that is in the form of the order of these nitrogenous bases. So this is the allele for purple color and this is the allele for white color. So what is the difference here? You can see it's only one nucleotide is different. So for purple color it is C and for white it is A. So this sequence, it codes for an enzyme. So in this sequence, a protein is formed which codes for the synthesis of the purple pigment. But because in this there is A instead of C, there is absence of the enzyme that can help in the synthesis of the pigment. So that is why these plants, they have white flowers. Okay, another important thing we need to know is Mendel actually gave two laws from his monohybrid cross, and they are the law of segregation and law of dominance. So basically, law of dominance says that one of the allele in the heterozygous situation, one of the allele is dominant, another is recessive. So the dominant allele, it expresses itself in the presence of the recessive allele. So you can see here, we just explained this. I showed you the sequence. So this is a dominant allele. This is a recessive allele. So in the presence of the dominant allele, recessive allele cannot express itself. Okay, it cannot express itself. It is the dominant allele which is going to express, because in this specific case, this sequence is going to code for a enzyme which helps in the synthesis of the pigment, and that is why the flowers, they have this color. So when this gene is there, when this allele is there, it cannot make the enzyme. If it cannot make the enzyme, it cannot make the pigment. So the color is going to be white. So he gave the law of dominance. Another law he gave was law of segregation. And the law of segregation can be understood here, that here, if you see here, he crossed two plants. One is purple, another is white. In the next generation, all flowers were purple. Okay. So here, what is happening is the white alleles, they're not getting mixed up. There's no blending hypothesis. There's a particulate hypothesis. Here, the white alleles, they are not getting blended with the purple alleles. But what is happening is the white alleles, they are not able to express, the white alleles, they are not able to express themselves in the presence of the purple alleles. So there's a law of segregation, which means these alleles, they do not get blended. They segregate. They get separated during the meiosis. They get segregated. They get these recessive alleles and the dominant alleles. They get separated. They get segregated during the meiosis. And that because they get segregated, they got separated during the meiosis. So that is why in the F2 generation, the white color comes back. It did not get mixed up. It did not get blended with the purple alleles. It got separated. It got separated, and that is why in the F2 generation, when both alleles were small p, this color showed up. So this is the law of segregation. Okay, here is a test cross. Basically, test cross is a cross by which you can know if the plant is heterozygous or pure. Suppose you have a plant with purple flowers. And the flowers are purple. So how to know if the flowers, they are pure purple, or how to know if they are homozygous or heterozygous? So how to know if the flower is both capital P's or one capital P and small p? So the way to do is do the test cross. So the test cross is a cross in which you will take a plant. You can take a white plant which is having both the alleles are recessive, and just cross them. So what is going to happen? Because if the plant is pure, which means both the alleles are capital P, when you cross this plant with this plant with small p's, then the next generation is going to be all heterozygous. The next generation is going to be all heterozygous, and all the plants are going to be purple. So if all the plants are purple, it means that this plant is pure. But if the plant is heterozygous and when it crosses with this plant, then 50% will be purple and 50% will be white. So if you get this kind of a result, it means that this plant is heterozygous. This plant is heterozygous purple. Okay, the third law is law of independent assortment, and that can be understood from the dihybrid cross of Mendel. So in the dihybrid cross, he actually studied two characters. So this is the dihybrid cross of Mendel. So basically in this, what he did, he took the plant with these peas. In this cross, he has studied two characters. So the first character is the color of the peas, and second is the texture. Okay, so he took a plant with round peas, and so they are round and smooth. Second plant he took with the peas which are green in color and they have wrinkled texture. So now, so this is a pea. These are peas. One second. Yeah. So remember that what is done. This is the P generation. This is a dihybrid cross. This is the P generation of dihybrid cross, and you can see here the plant he has taken. Sorry, the peas he has taken. So these peas are basically the seeds. Okay. So the pea plant has flowers and it has the peas the sweet pea. This is sweet pea. So they are in those pods. So these are those peas basically. So he has taken one plant which has peas with yellow color, and yellow is a dominant character, and its texture is round, and round texture is also a dominant character. And other plant he has taken is with the other plant he has taken is with the green color. The peas are green color, and the texture is wrinkled. Okay, so both plants are again pure for the characters. Now remember that when this plant is going to make the gametes. So I think we need to go to the board again and we need to see that. I have to explain this again. Okay, so let's erase this. Okay, so when Mendel took in the dihybrid cross, when he has studied two characters, important thing to know about these two characters is that these two characters, the alleles for these two characters, they're present on two different chromosomes. Now, if suppose this is first parent, okay. Okay. So the two alleles, the alleles for these two different characters are present on two different chromosomes, two different chromosome pairs. So this is first chromosome pair. Okay. So here is the alleles for the color of the peas, and this is first chromosome set. This is the second chromosome. These are homologous chromosome. This is second chromosome set, which has the alleles for the texture. So this plant has both alleles, capital R. So this is one parent. Okay, now second parent. Now in the second parent, this is first chromosome pair, this is the second chromosome pair. Okay, so this is this chromosome has small y and small y. Okay, so the allele for the color of the peas is small y and small y. These are pure plants. Okay, and this plant has a second chromosome set where the alleles for the texture are there, and that oh, I'm so sorry. No, I'm right. I'm sorry. So the texture is small r and small r, which means they are wrinkled. Okay, so this is plant number 1. This is plant or parent number 2. Okay, so this parent genotype is capital Y, capital R, and capital R. This parent is also pure. Its genotype is small y, small r, and small r. Okay, so small y is for green color, which is a recessive character. So these alleles are on this chromosome, and small r, these are the two alleles for the texture, and they are recessive characters. These are the wrinkled, that's for the wrinkled texture. Okay, so this is the genotype of this plant, and this is the genotype of this plant. Now when the meiosis takes place, it is going to be the separation of the chromosomes. So in the meiosis, these chromosomes are going to separate. In the meiosis of this plant, these chromosomes are going to separate. Okay, so in the meiosis, this plant is going to make all the gametes capital R. All gametes are going to be like this. For this plant, all the gametes are going to be small y and small r. All the gametes are going to be like this. Okay, when this plant fuses with this plant, what is going to happen? The next generation plant is going to be capital Y, capital R, and small r. So this is going to be happening in the F1 generation. So because capital Y is a dominant color, which is the yellow color, this plant is going to have yellow color, and capital R is for the smooth texture. This plant will have smooth texture. So in the F1 generation, all plants will have this genotype. All plants will be yellow and smooth. Okay, this is the progeny of F1 generation. Now let's see what happens. Now if you have, so in the dihybrid cross, he took these plants. He took the two plants with this genotype, and he crossed them. So to understand what will be the next generation, we need to understand how the gametes are formed, right? Again, now let's make, okay, so this is chromosome pair number 1 for, okay, so remember that these two characters are on different chromosomes. Now this is the second set. Okay, okay, so here is capital Y, here is small y, right? For the texture, they are heterozygous. Capital R, small r. For this plant also, capital Y, small y, capital R, and small r. Okay, now we have to see what kind of gametes they're going to form. Okay, now suppose we have, now this is a case of heterozygous, this is a heterozygous situation. Now remember what we talked in the meiosis, and this is a heterozygous situation, and also in this case the chromosomes involved are more than one. Unlike in monohybrid cross where there was only one chromosome involved. In this case, there are two chromosomes involved. Now, in this case, you can see that if suppose, remember, go back, check what happens in the metaphase. How the chromosomes are aligned, it is independent of the other chromosome. So, how this chromosome is aligned, it is independent of this chromosome. If capital Y is on this side, okay, if the chromosomes are aligned like this, the way these are. So when they separate, what is going to happen? The gametes which will be formed will be capital Y, capital R, and on this side is small y and small r. This is the second gamete. First gamete. But what will happen? If the orientation was different? Which means that suppose the orientation was like this. Suppose small y is on this side and capital Y is on this side. Then what will happen? The gamete which will be formed in this orientation, will be small y, capital R. And this side will be capital Y and small r. Okay. Now you can also switch this R on this side and this R on this side, and you will see that in total these are the 4 kinds of gametes which are going to be formed. So in short, we can also, in a simple way, I can also tell you that if your genotype is like this, okay, this capital Y can go with capital R, then the gamete is going to be capital Y, capital R. This capital Y can also go with small r, then the gamete is capital Y, small r. This gamete can go with this, Y can go with this. Small y can go with capital R. This small y can also go with small r. Small y, small r. So these are the 4 gametes possible from one plant, one parent. Okay. These are the 4 kinds of gametes which are possible. And so what he did, he took these plants, these are 4 gametes possible, and he did crossing off the F1 generation, and then he formed the F2 generation. And in the F2 generation, the Punnett square is like this. Now because there are 4 gametes, there are 16 offsprings possible. So you're going to make Punnett square like this. Okay, so you're going to write the gametes of one parent. So one parent is male, another is female. So capital R, capital Y, capital R, capital Y, small r. Then it can be it can be capital Y, capital R, or it can be small y or small r. Okay, so capital Y can go with capital R. So capital Y, let's do like this. This is capital Y, okay? Capital Y can go with capital R. Capital Y can also go with small r. Small y can go with capital R, or small y can also go with small r. The same thing here, second parent. Capital Y can go with capital R. Capital Y can go with small r. Small y can go with capital R, or small y can also go with small r. Okay, and then they can fertilize, and then you can see if this gamete fertilizes this, the progeny will be capital Y, capital R, and capital R. And this is the way all boxes you can fill. Okay, so on the basis of that, we can detect the characters. Now this is going to be yellow and round. So if you go back to the PowerPoint, you can see here, this is what the dihybrid cross is. This is what I explained. So this is the plant, the parental plant, these are the gametes, and this is the F1 generation. And from this generation, from the F1 generation, these are the gametes formed, and when they cross, they form this. So when these, I just wrote down these gametes on this side, these gametes on this side, and you can see when they fuse with each other, they form these kind of progenies. So they form these different kind of characters. The plants can be having yellow and smooth seeds. They can be having green and smooth seeds. They can be having yellow and wrinkled seeds, or they can be having green and wrinkled seeds. And the ratio is of all these different kinds of seeds is 9:3:3:1. So which means that out of 16, 9 plants will have yellow and round seeds. 3 will have green and smooth seeds. Another 3 will have yellow and wrinkled seeds. And 1 is going to be green seeds with green and wrinkled seeds. This is the dihybrid cross, and this on the basis of this dihybrid cross, Mendel gave the law of independent assortment. So he said that these characters, they are not dependent on each other, which means that as he took the yellow and round seeds, and here he took the green and wrinkled seeds. So the inference or the insight he got from this cross was that he said that these characters, they are assorted independently of each other. The reason was because he could see the yellow color. He started with yellow with round texture, but he also got yellow with wrinkled. He started with green with wrinkled. He also got green with smooth. So, which means that all these characters, they are assorted independently of each other. So, the division meiosis came later. People came to know about this division later, and then they could actually see the basis of what Mendel could see here. Okay, so now the way I have explained you in terms of chromosomes, that understanding we have now, because now we understand meiosis. Okay, at that time, this is what he got. This is all the characters of plants he got, and he made an inference. But what is the basis of that is the cell division meiosis, and the basis is how the chromosomes they are getting, how the chromosomes are separating, how they are sorting independently of each other. So this is what is on the basis of that he gave the law of independent assortment. Okay, I'm going to stop this video here. This is all Mendelian genetics. In the next video, I'm going to talk about the non-Mendelian genetics.